Okay, I hope that you're excited because now we're going to see what the view looks like. This is my favorite part of this tool. And I think that the view we provide of the alignment here at BV-BRC is pretty awesome. So I'm going to navigate to my jobs. I generally go down here, but like I told you before, I could go up here and go to my jobs, and I know what this one is called. One thing, let me point out, like if you want to filter on things and just see, oh, I want to see all the genome alignment jobs, you can do that, and then it's genome alignment all the way down. But this was the job I first ran with you guys. So I highlight that, and when you click on something, it gives you three possible things from the jobs page that you can do in the green action bar, which was rerun, report issue, or view, which is the big eyeball. So you click on that. We already talked about all the files that are available in the job results landing page. What we didn't do is click on this mysterious view icon in the upper right. So let's do that. Now, depending upon how large your genomes are or how many of them you submitted, this can take a while to load. So just wait, it's going to come. All right, so here it is. And I think this looks very nice, even if I do say so myself. Biologists generally like mauve and they like this view. So let's talk a little bit about it. These buttons at the top are some of the controls. After you've messed with it, you can reset the display. You can go into options. And before I do that, you notice here it's got the name of the genome and then the unique identifier, which is the genome ID. And here's the second one that was submitted and the genome ID. And the third one in the genome ID. If for any reason, like let's say you're going to publish this thing, you don't want the genome IDs in there, you can click that off and they go away. You can click on options, and these lines that are connecting the blocks, I can click on that and they go away. So let's say you didn't really like the way this line was going, you could get rid of those or add different ones in a different, I use Adobe Illustrator so I could manipulate it if I wanted it to look a little bit different. At some point, we're going to pretty soon show you how to see the features when you zoom in so you can remove that if you want. And then you may be wondering, what are these red lines that are going down? Those are the contig barriers. These Brucella are complete genomes, and Brucella has 2 chromosomes. So from here to here is chromosome 1, where this red line comes down, and from here to here is chromosome 2. So I could say, I don't want to see those contig boundaries, and you can make that go away. And this might be good to do, like if you have one that was in hundreds of contigs and it was all little red marks, although that would not look good anyhow, but just giving you the options for that. So you can zoom in even to the level of the genes. So let's just see what that looks like. I just keep clicking on this icon. Ding, 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 ding. Ah, and then I mouse over that and it tells me what that gene is. Then if I don't want to do that, I can mouse back. And if I want to scroll one way or another, I can do that. Notice when I'm going over this, these are the blocks. Mauve has identified these as highly conserved blocks with that seed weight, which was the default seed weight, which we used was 15. So when it was set at that, these were the blocks of similarity on the 3 genomes that it found. And so this is the first one. And notice above where it says position, LCB length, contig, and contig length. It's telling you information about this one. Now look what happens when I go down below. There it changes because we're in Brucella suis now, not Brucella melitensis. And then here it changes yet again. And as I mouse over it, the position will change, but the LCB length and the contig don't change until I get to here. Now notice when I moused over that, that light blue line between the blocks is highlighted. That's the LCB line. That's saying these guys are shared. And then we get into the contig here, and then this block. And if we go up to the top one, then we have this block, and look what's happened there. In melitensis, this block of the genome is towards the beginning of contig 2, but in suis and abortus, the second and third ones, it is at the end. So something has happened here. There's been a genome rearrangement. And then you can see this one, and look what's happening in abortus here. This particular block is above the level of every other block in the genome. This is an indication of an inversion. And then the final block is showing that in melitensis, this is at the end of the second contig, and here it's right early on in the second contig. So let's say you really like this thing, but you wished you'd set it up differently. You wish that abortus would have been here. That's what you really wanted to see. Oh, do I have to run the job again? No, you don't. You can just click here and it moves it up, and then you can have the view that way. And when you get it just the way you want, you can click on the SVG, scaled vector graph. So I click on that, and it would start to download that image, and it will be in your download file. And then you can upload it into Illustrator or submit it to a journal if they like the SVG format, and you're good to go. And I think these look really good. So I would encourage you to use those SVGs. I want to scale in one more time. So I'm going to scale in here, and I'm going to walk down the genome as I scale in. And get in, because there was one more thing I wanted to show you on this. So you notice when I mouse over these things, it shows me the names of the genes and that information. So I want to get right to a boundary, like right here. So let's say I'm really interested in what's happening here, this particular protein, and that's the one. Oh, this is at the contig boundary. I really want to see that. What do I have to do? It tells me here the unique identifier for that particular gene, which in BV-BRC is the one that starts with FIG, and then there's the line, and the next one is the genome ID for that genome, which is 224914.11. And then it says PEG, protein encoding gene. And then 2370, that's the 2370th gene in this genome. After that, it gives you the GenBank accession for that gene. And then it tells you what it's called and its location, where it says CDS 92332 to 93318. That's telling you the start and the stop of that gene. But let's say you want more information. So double click on that and everything you wanted to know about this gene. And if it's blue, these are hyperlinks. So you can click on that and it will take you to RefSeq or take you to GenBank. Here's the genome. Here are the protein families that this is in. It tells me that it's part of a subsystem, so it's a gene of interest because it does something interesting. And if I wanted to go to the gene landing page, I could click here. And this is one of the reasons why we want you, or we demand that you annotate your genome in BV-BRC, because we provide this type of integration for you so that you can see these things and see the information about it. And then you could see which subsystems this was involved in. Also proteins, things that have the identical protein sequence or gene sequence. So if you click on that, it'll show you how many genes in the system have an identical system. And this guy's kind of unique. It doesn't have too many of them. So those are the kind of things that you can do when you're in the system, when it is annotated, and that's why we want you to do that. Now, in the next video, we're going to talk about what happens when you start adjusting those things. Remember when we clicked on the advanced parameters and I showed you that you could adjust the seed weight and the weight? I'm going to try to show you some of the things that happen when you do that. So I will see you then.